Welcome, everybody, again. Bringing you another really awesome show today. Today, unique show. We are hosting not one but two of what could only be described as the founding fathers of this modern longevity geroscience movement that we find ourselves in 2024. And while they may have come at the issue from different angles over the years, and many have always wanted to see a debate between these guys, and we maybe do a little of that. I think their approach is, while scientifically, from the basic research side, a bit different. They've converged quite a bit over the years from the translational angle, more than some may realize. First, we're honored today to join me Dr. Aubrey de Grey, President, Chief Science Officer, Longevity Escape Velocity Foundation, which is focused on proactively identifying and addressing challenging obstacles on the path to the widespread availability of effective treatments to prevent and reverse human age-related decline. He's needlessly internationally recognized as a visionary biomedical gerontologist. He devised the strategies for engineered negligible senescence, the very comprehensive set of methods that he promotes to rejuvenate ultimately the human body, preventing age-related ill health and mortality, and has founded multiple organizations, including Methuselah Foundation, SENS Research Foundation, and now LEV, or Longevity Escape Velocity Foundation, to enable this research and to accelerate it. We're also joined by Dr. Michael Rose, Director of the Network for Experimental Research on Evolution, as well as Professor in the Department of Ecology and Evolutionary Biology at University of California, Irvine. There, his primary work over the years has been on the evolution of aging, and in 1991 published seminal work per the Evolutionary Biology of Aging, exploring the topic of antagonistic pleiotropy, the really novel hypothesis that genes act at different points in life and may have different effects, and ultimately, sort of as he will define, the inability or the declining forces of natural selection ultimately lead to what we see as age-related pathologies. He has, he's also extensively published over the years, including authoring The Long Tomorrow, How Advances in Evolutionary Biology Can Help Us Postpone Aging, and is also, has been conducting one of the longest-running artificial selection experiments per the antagonistic pleiotropy concept in fruit flies, which started back in 1981, where he quadrupled the lifespan of these animals. We'll be getting into a lot of really interesting themes today. Honored to have both of them with us. A lot to get into. Dr. Aubrey de Grey, Dr. Michael Rose. It's great to have both of you on the show at the same time.
Well, thanks
Hi,
for having me.
Ira.
Good to see both of you. A lot to get into. I thought, we could start off, I guess, first with Aubrey. A couple months ago, Aubrey, you publish, I guess, your most recent paper. It was entitled "The Divide and Conquer Approach to Delaying Age-Related Functional Decline. Where Are We Now?" Where you look back at the last 20 years. You talk about sort of the big picture themes that you've seen in terms of, we got this complex thing called aging. There's not going to be single magic bullets for it. If we classify the types of damage that make up aging, we better classify type of repair processes to interfere, or we don't have translation, we just have more research funding. And lastly, the combination rejuvenation studies, while something that's been neglected, or something that we need to see a lot more of as we move further into the translational space. Talk a little bit about this paper. Take us back a little bit to the last 20 years, and a little bit up to, some of the core learnings, and don't go into robust mouse rejuvenation yet. That'll be coming up in a little bit.
Yeah, sure. So, yeah, so 20 years is a long time. Excuse me. And it was actually just on 20 years ago that I founded the journal Rejuvenation Research, where I published that little commentary at the end of last year. And so really what Irina Conboy, the current editor-in-chief there, wanted me to do was to indicate, how things have changed and progressed. And I think a lot of the focus I had in that paper was on the fact that what I said 20 years ago has very substantially stood the test of time. And thus, that, of course, it has a good chance of continuing to stand the test of time. Some of the stuff that I talked about in that paper related to the, well, comparing and contrasting the original characterization of aging that I put forward back in the year 2000, 2001, 2002, with the hallmarks of aging, the famous essentially restatement of the same concept that came out a decade later. And the reason I wanted to do that was, first of all, to emphasize that, there's no real debate about this anymore. People don't really think in terms of theories of competing theories of aging based on hypotheses that this or that single unitary mechanism is, dominant in aging. There's very little of that these days. There's much more of a general acceptance that aging consists of a a set of only loosely cross-talking processes of change to the structure and composition of the body that are eventually pathogenic. And the way that this was put forward in the Hallmarks paper, and of course in subsequent revisions to it and improvements to it, is largely what I said 10 years earlier, but there are some imperfections in the more modern versions of it. In particular, there is not nearly enough emphasis, in my view, on the reason why a classification is useful in the first place, why it's useful to take the many, many, many types of damage that accumulate in the body and partition them into this manageable number of categories. The actual reason why it's useful is because the classification is designed to allow a one-to-one correspondence, or one-to-many correspondence actually, between categories of damage and ways in which the damage could be actually repaired and eliminated from the body, which is what I have always called rejuvenation. Back 20 years ago, there was really only one plausible, in my view, way to do each of these things for each of the categories. Now there's multiple ones for each of them, which is nice. But the idea of thinking that way at all was, of course, very heterodox back in the year 2000 or so. And in fact, the meeting at which I had the eureka moment that encapsulated and enshrined this idea was one in LA where Michael was present in the year 2000. And I had this idea in the middle of the night during the middle night between the 2 days of this meeting. I came down the following morning and presented it to everybody, and I didn't do it very well, and nobody understood a word I was saying, but that gradually changed. So that's really what the little article I wrote last December was focused on.
Excellent. And Mike, we talked a couple of years ago now about your work at the time, discussing obviously your seminal book from back in the early 90s, about declining forces of natural selection and antagonistic pleiotropy and your fruit fly experiments. And about the time you had published this chapter in the Transhumanist Handbook entitled "Are You Willing to Die for Reductionism?" And this chapter sort of gets into a lot of what we were discussing on the show at the time, that based on your extensive work in your fruit fly experiments, that no, there wasn't one gene or two genes or three genes that were causing this this amazing sort of difference between the short-lived and the long-lived flies. But as you were pointing out when we were chatting, hundreds, if not thousands, of sites across the genome involved in what you were looking at per your concepts and your theories as aging. Take us up to reintroduce us to your work a bit and take us up to sort of that 2016 chapter, if you would. That puts us at a sort of an important place where we'll follow up afterwards.
Well, thank you, Ira, and thank you for hosting this discussion between Aubrey and myself. Aubrey and I share the exact same goal. We only ever differed on means and context. So I'm actually sitting here primarily representing a field. And in many ways, Aubrey is the tip of the spear of a different field. So there's the cell molecular tradition of gerontology, which actually has its roots in the core hypothesis of Aristotle, that aging is a cumulative physiological process of some type of disruption, disharmony, or damage. And that has been the overwhelmingly predominant view of aging throughout Western civilization. And right now, because cell molecular theories of causation are dominant in biology, these days, the flavoring of that basic cumulative physiological process theory of aging is phrased in terms of the hallmarks of aging. I represent a dissenting tradition that, of course, was founded by Charles Darwin. August Weismann, in the late 19th century, made the simple point that if we have immortal germlines, evidently, if natural selection wants to produce immortality at the level of cells, organisms, it can easily do so. I don't know how aware he was of the existence of biologically immortal animals and plants, everything from trembling aspen to fissile sea anemones. Fissile sea anemones, in particular, having similar cell and molecular biology at the level of the cell to ourselves, do not undergo any aging process whatsoever. And that core insight has been developed over the last 100 years. And basically, if you regard natural selection as the ultimate source of everything that works in biology, as opposed to all the stuff that gets screwed up, then the question is, well, if natural selection can clearly produce biological immortality, where your main risk of death is infection or mechanical accident, then why hasn't it bothered to do so in a whole number of very prominent cases? The one we care about most is humans, but in fact, aging is universal among all mammals and all insects. And it turns out, as with evolutionary theory in general, we have a physics-like body of mathematical theory, and that body of theory started to be applied to the problems of aging clearly in 1966 by William Hamilton, then all through the 70s by my doctoral advisor, Brian Charlesworth. And basically, unlike the diverse cell molecular mechanisms of the cell molecular tradition, evolutionary biologists see one core cause of aging, which is the declining forces of natural selection. And we have not only pointed to comparative evidence that this is fundamentally the correct scientific theory of aging, but in my own lab, I have exploited manipulation of those forces of natural selection to change when aging starts or stops in Drosophila. Yep. And a very non-trivial aspect of the evolutionary theory, which is my main focus of academic research for about 30 years, is the cessation of aging, which is something that aging can do entirely on its own and can be manipulated by manipulating the forces of natural selection. Now, I've, together with my graduate student, Kenneth Arnold, done the world the favor, I think, of compressing the last hundreds of years of research into one relatively slim book called Conceptual Breakthroughs in the Evolutionary Biology of Aging, available now on Amazon and very few other places. So effectively, the evolutionary biology of aging has reached a point of maturity and power. And the power is that we can produce organisms that live much longer. We can produce organisms that stop aging at different times, or that don't start aging for what used to be deep into their lifespan. And therefore, technologically, having done that, we can look at, well, what are the cell molecular mechanisms by which you produce a much longer lived organism, which is the goal that Aubrey and I share with respect to humans. And the answer is on the order of 1,000 sites in a fruit fly genome, a number that's unlikely to be lower in the human case. on the order of 1,000 transcripts at the level of the transcriptome, which includes all the influence of DNA methylation. And in terms of metabolites, almost certainly tens of thousands of metabolites. We're working on estimating that number, but the number is on the order of magnitude of half of all the metabolites have to be retuned. This would include all the products, all the kinds of damage and disruption that come from cell molecular metabolism. So rather than having to address, the physician's favorite 20 rogue metabolites associated with cardiovascular disease or Alzheimer's disease, etc., our very powerful experimental systems show quite clearly that you're looking at having to deal with tens of thousands of pathways, which crisscross from genome to transcriptome to metabolome. And furthermore, we can use tools like gerontology, and we can also use machine learning to connect those thousands of pathways at whichever of those levels you like to investigate to actually organ damage and failure. And we've made a lot of progress in, our toy model system of the fruit fly. And it's all effing complicated. And it does not conform to practically anybody's idea or a priori conception of what aging should involve. It turns out that organisms don't read the papers of some molecular biologists. They don't favor the kinds of hypotheses even that evolutionary biologists were thinking of in the 1930s and 40s. The omic phenomena and indeed causes of aging are extremely complex. So the question becomes, to get back to the goal that Aubrey and I share, what do we do about this?
Yep.
And I think the organisms have to tell us. And because we now have spectacular power at resolving what the genome does, at resolving what the transcriptome does, at resolving what the metabolome does, we even have far greater power at working at organ and system level physiology for organisms like Drosophila, nematodes, and humans, the three main animals of biomedical research. I think as we progress past the point of scientific debate, to my mind, there is no scientific debate. There is a unitary cause to aging. It's the forces of natural selection giving up on indefinitely sustaining the adaptation of humans and fruit flies. Now we're really in an era of technology.
Yep.
Now, the exciting thing about the meeting that Aubrey and I were both participants in, which is a very small meeting, was a meeting very much about turning from science to technology. And I think that this is the key moment where, the scientific debate is over. As Aubrey said earlier, every unitary cell molecular hypothesis is dead as a unitary hypothesis. There's only one remaining unitary general field theory of aging supplied by evolutionary biology. If you want to know the science about evolutionary biology, my colleagues and I, we produced hundreds of publications and, a handful of books. You can go read that stuff. The question is, given that we have complexity, what do we do with the complexity?
Right.
And my favorite approach is to do something like what the Wright brothers did, which was do simple model system work. I think right now, trying to answer really hard questions using the very limited genomic resources that we have for mice isn't powerful enough. I think fruit flies and nematodes are the two most powerful systems. Nematodes are fantastic systems for understanding the problems and possibilities of inbred mice, which is most of the mice we have. Not all, but most. I think Drosophila is the best possible system for understanding what relatively outbred species, which is what humans are, what their aging looks like. Dogs are kind of intermediate. They're not as inbred as inbred mouse stocks. And if you're very concerned about your pet dog, you should be very interested in nematode research, NIH-funded mouse research. And I think that's a key to the quite strikingly inbreeding-related health problems of dogs. Cats are feral relative to dogs. Cats are more like humans in terms of their underlying genetics and genomics. So if you're a dog lover, NIH and nematode research is your key to unraveling the mechanisms, the machinery, and the technological possibilities of enabling, your pet dog, Fluffy or Rex, to live longer. Unfortunately, we are not remotely like nematodes, inbred dogs or inbred mice whatsoever. We are not even the products of multiple cross inbreeding, which some of these so-called outbred mice are, which are quite novel and bizarre products of genetics. Now, not all fruit flies are outbred. The kinds that the evolutionary biology of aging works with are usually outbred. So I think outbred model systems are the systems to go. If your concern is humans, as opposed to dogs or inbred mouse stocks of the NIH. So, and I think we've made a lot of progress with Drosophila. And I'm thinking that this is sort of the last decade where Drosophila will be at the frontier of solving the technological problems. I am hoping that this decade marks a transition for people like myself, okay, and other people who work with outbred animals, to make a transition from working with things like outbred lab fruit flies to outbred human patient populations. And I think the key to that is the fact that so much of the Drosophila genome and transcriptome is very, what biologists call homologous, what everybody else would call similar to the human genome and the human transcriptome, and even a large proportion of the metabolites, of course, by no means all, of the basic housekeeping pathways of fruit flies are in common with humans. And companies that I've been involved in have we've published results where we've shown that the Drosophila omic results do in fact map into human omic results quite powerfully. Mm-hmm. And this is a staple in the world of genomics, this mapping from one species to another. And that's the greatest hope, I think, for mastering the enormous technological challenges that we face. And I would really like to thank Aubrey for that day in Manhattan Beach, the founding of the Manhattan Beach Project, where he said, okay, I'm tired of all your arguments about this theory or that theory. Let's focus on technology. Let's regard this as an engineering problem. And I completely agree with this engineering focus, with this technological focus, with leaving the scientific debate behind, because in my mind, it's over, and mastering or exploiting our mastery of all of the omic and machine learning tools required to deal with complexity in the complexity of aging in an open-minded way. So, to be concrete, maybe 2 or 3 of the hallmarks of aging are in fact causal. Okay, they are certainly hallmarks of aging. It's stuff you can see. Stuff you can see isn't necessarily causal. This is a core lesson of science, right? So if 2 or 3 of those hallmarks of aging are key to the actual physiology of the entire organism that is a human and its aging process, and we find an intervention for that hallmark, and has causal benefits, fantastic. However, our research unequivocally shows there's so many more things going on beyond cell molecular hallmarks of aging. And whatever those are, I think we need to intervene with those.
And I think you both are, as again going down that translational path again in different ways, but again, you're both again championing sort of our next step, which is extremely important. Aubrey, let's focus back on you now in terms of where you've gone with robust mouse rejuvenation. We talked about 8 months ago or so at the beginning, and clearly, obviously a lot of what you worked on per the seven SENS spun off into different companies and initiatives, but core to what you're doing now per LEVF is this combinatorial robust mouse rejuvenation. Could you walk us back through that story and a little bit about what's happened in the last 8 months? Because there's been a lot going on. I've been, watching your monthly updates on social media.
Yeah, I think I'm going to have to go back a lot further than that in order to give any kind of context.
Sure. Take your time.
So let me do so as best I can in the context of everything that Michael just said. Michael and I are often characterized as disagreeing a lot, and I think, it's very important to understand that the main thing we disagree about is how much we disagree about. In other words, I think we disagree about a lot less than you might have inferred from what Michael said. The main thing I disagree with Michael about in that sense is the idea that there is some kind of conflict between the evolutionary viewpoint of aging and the mechanistic reductionist viewpoint. I believe that they are not in any way in conflict. They are just two different descriptions of what's going on. At a very simple level, one could say evolution is the evolutionary viewpoint is the theory of, the body of theory of why we are what we are biologically. And the reductionist approach says what that translates into in terms of how we eventually go downhill as we get older. And so I don't think there's a conflict there at all. I think that the work that Michael's done over the past 40 or more years is among the most profound and fundamental bodies of work confirming and of course elaborating the model of how evolution does what it does in relation to aging. But at the end of the day, what evolution does is it gives a new organism its genome, and it then that organism does what it does with that genome. The genome determines what happens next. It determines how rapidly things go, for example. One thing that I also want to highlight here with regard to where Michael and I agree is the complexity of metabolism. I believe that wonderful though her contributions were over the years, especially in the 1990s, Cynthia Kenyon did something of a disservice to the field by oversimplifying the way that we look at metabolism, the way that the field and people outside the field especially look at metabolism. Because what Cynthia did, and of course Tom Johnson a few years earlier, was to demonstrate that single gene modifications in a simple organism, the nematode worm, could elicit very substantial increases in longevity. And this, when Tom did it, was so antithetical to what everyone had decided, which is essentially what Michael just told you all, that everyone basically ignored him. It needed to be done again by somebody else independently before anyone really paid attention and believed that it was true. But we should have understood at once, really, that this was something of a false dawn, because what was happening there was a genetic emulation of something that is not in any way of the character that Michael just described, this enormous complexity. Rather, it's a single thing, the response to famine, essentially, the response to calorie deprivation. Of course, the idea there was that this was genetically phenocopied. It was, these genes essentially tricked the worms into thinking they weren't getting enough calories and caused them to activate parts of a pathway that they naturally have in response to actual lack of calories. Now, this same thing, of course, has been done genetically in mammals, in mice, and in more recent years, it's been done pharmacologically in mice. But the point is, it is the elicitation of a programmed response, something that evolution has put in place as a way to respond to variations in the environment that are seen often enough that evolution actually paid attention. And this has the rather unfortunate corollary that the whole thing doesn't work so well in long-lived organisms as the way it does in short-lived organisms, because essentially long famines are not so frequent in nature as short famines are. I was so shocked back 20 years ago that people were sweeping this under the carpet that I actually wrote a whole paper about it. But that's been published, obviously, so you can look it up later. Point is here, metabolism is insanely complicated. And if you want to properly mess with metabolism and make it do what you'd like it to do to make us age more slowly, if you like, make the body run more cleanly, then, good luck to you. It's just not going to happen. Metabolism is far, far too complicated, far too many different pathways, far too many interactions, far too many unintended consequences that will happen if you try. You basically just can't disentangle the things we don't want metabolism to do, namely make us biologically older, from the things we do want it to do. And this is essentially why, before Cynthia came along, everyone had given up. And it had become completely, unacceptable to talk about intervening in aging in a grant application, for example. So essentially, Michael and I believe, have two different but equally valid ways of sidestepping this complexity. And both of them, excuse me, and both of our ways rely on recognizing that biology actually wants organisms not to age. It just doesn't want it very much. So essentially, what we have with Michael's approach, as I would say it, in putting it in my own words, is if evolution is given more of a reason to postpone the physiological decline of late life, it will respond. And what I say is that we have this rather convenient fact that the things that go wrong with us late in life also sometimes go wrong with us earlier in life in isolation as a result of infections or injury or whatever. And the body has mechanisms that work quite well to restore the body's function earlier in life. Many times we may be able to elicit that same thing. In other words, again, we are on the same side as what the body's on. I'm now talking about how the body responds to mechanistically, as opposed to what's in the genome, rather what results from what's in the genome. But it's still the same thing. It still means that if, rather than trying to tell metabolism what to do, we just make it easier for metabolism to do what it has evolved to do, then we've got a chance. And this is essentially the entire eureka moment that I had in the year 2000, that if we can restore the structure and composition of the body at the molecular and cellular level to something like how it was in young adulthood, then we will automatically restore the function as well, including the remaining longevity, just because the body's a machine. It's an insanely complicated machine. We don't understand it. We can't manipulate it properly, but it's still a machine, which means that its function is determined by its structure. So the more we can restore the structure to how it used to be, the more it will restore its function to how it used to be. Therefore, I don't think there's any conflict between the evolutionary viewpoint and the mechanistic viewpoint at all, just so long as one describes the mechanistic viewpoint in the way that I do, in terms of preventative maintenance, rather than in terms of making metabolism run more cleanly, which was the prevailing paradigm before I came along, if you like. Mm-hmm. Mm-hmm. Right, so now, Ira, to come to your question, what are we doing about this? Well, any kind of damage repair, preventative maintenance approach, whether it's for a human body or for a car, is a divide and conquer strategy. you've got to develop a whole bunch of different ways to repair different types of damage. And I mentioned the utility of this classification that I put forward in the year 2000. That is simply to essentially get our arms around the process of devising these interventions, because within each category, we can talk about a generic damage repair approach, which differs a little bit in the details from one example to another within the category, but which also, every example has a lot in common. So it means that the whole thing becomes more tractable. And so you've got to develop these things, but then you've also got to put them together. So I think of this as a two-stage process. And stage one is developing the individual interventions to repair individual types of damage, and then combining it in the same organism at the same time and identifying all the, unforeseen interactions and so on. So at SENS Research Foundation, my focus was on phase one, especially on the more difficult ones, the more difficult types of damage that were being ignored by most researchers who were in academia and had to get their next papers out in order to get their next grant application funded, or in industry where they needed to get profits quickly. I was able to escape all of that short-termism by being funded entirely from philanthropy. And it worked well. And as you mentioned, Ira, we were able to move half a dozen projects along to a point where they did become investable and were able to spin them out as startup companies. But at LEV Foundation, we're moving on to phase two, which is the combining of the things.
Right.
And in particular, right now, we're doing an experiment just combining four things. These are four things that we would never have worked on at SENS Research Foundation because they're at the easier end of the spectrum of types of damage and types of damage repair, the ones that were already being pursued by other people. But the point is they'd already got to a point where they were working modestly well individually in mice. So you could take middle-aged mice and give these things, and the average and maximum lifespan would be somewhat increased. And our goal in this experiment that we're doing right now is to combine them and see whether we can get a considerably greater increase in lifespan. Everything's going pretty well so far. We're only a year in, so we started at 18 months. These are black 6 mice that normally live to about 2.5 years, so just on half of them, just over half of the controls are now dead. And I'm very happy to say that only about a quarter of the mice that are getting all these treatments are dead. So things are going pretty well so far, but we will see how long that lasts. In particular, we have a bunch more interventions that we'd like to combine, and we want to do follow-up, similarly designed experiments. But the experiments are very expensive. That's another reason why nobody else has done them. This experiment is 1,000 mice, so it costs, $3.5 million, because especially because some of the treatments, in fact, most of the treatments are not just what you put in the chow. They are like, injections of stem cells or genes or whatever, which is something that other people, such as, for example, the NIH's interventions testing program, have shied away from on the basis of reproducibility or whatever. Yeah, so that's basically what we're doing right now. Actually, I want to come back to one other thing that, just a very brief digression.
Sure.
One other thing Michael touched on, which was dogs. Because, there is work being done in dogs in the community, which are not so inbred. Of course they are somewhat, but not all of them. And the most important project, in my view, in that regard, the dog aging project run by Matt Kaberlein, has just been defunded by the NIH. It's absolutely disgraceful. It's like I've got absolutely no idea what's going on in the heads of people on study sections. But of course, that's why I have, as I mentioned earlier, constructed my work in this area so as to avoid the tyranny of study sections.
Yeah. No, I appreciate you bringing that up. That was a bit shocking the last couple of weeks, and I'm going to come back to you, Aubrey, and talk a little bit about, because I know there's some teaser stuff that you put out there on robust mouse rejuvenation part 2, and we'll want to get into that when we look a little bit more at the future. But going back to Mike, interestingly, again, since we last talked, again, I've been looking at a lot of your papers. You've been doing, as mentioned, you continue some of the fruit fly stuff in terms of the genomics work and the drug screening, but there was, several papers that have popped up, and again, this kind of, in some ways, different, but kind of similar in the on the topics of this um, evolutionary biology of nutrition, looking at interesting combinations of diets and botanical supplementation per your screening program. And again, when I think of things like that, specifically diet and botanicals, we're not talking about single magic bullets here anymore. We're talking about really interesting bioactive combinations of drug moieties or natural moieties that we have been co-evolving with over the years. Say a couple words about what you've been doing on the screening front, sort of traditional screening using the model system you developed, and a little bit of what you're doing, especially per your paper, notes towards an evolutionary biology of nutrition. I think this is really fascinating new stuff that we don't normally hear about per your program.
Uh, well, thank you for that invitation to discuss work that I'm excited about. The evolutionary biology of aging is not just a sort of background why, it's also the details. And one of the most important details being that aging stops. And I'll just make a asterisk under something that Aubrey was saying, in that he was referencing the young organism. For me, the most interesting organism is the organism that has stopped aging, which in fact people on the standard American diet do after 105, which is tragic. But people on the organic agricultural diet that Europeans had prior to 1930, humans stop aging at 90, which is very promising. This then relates directly to the question of nutrition. We get a 15-year difference in when aging stops, and that's pretty radical in terms of the human lifespan. And that was sort of stuck in my head for years. And then in around 2009, 2010, I realized that there was an aspect of the mathematics of the evolution of aging that everyone had neglected, me included. And that is that the forces of natural selection don't just scale aging as a decline in adaptation across the entire metabolism. They also scale how rapidly an outbred species can adapt to a dietary change.
Right.
So we've gone through 3 titanic dietary changes over the last 1.5 years. 1.5 years ago, we were transitioning from a chimp-like sort of adventitious scavenging, gathering, feeding on fruit and grubs and other things that were relatively easily obtained, into being first hunter-gatherers, where we developed weapons with which to kill game, and also to scavenge better by cracking open brain cases and bones to get at brain and marrow, to foods that are full of nutrients for evolving a larger brain, which we then did. And we started cooking our food. So somewhere on the order of 1 million years ago, we were omnivorous. We were extremely well nourished on foods that we hunted or gathered from our environment, that we processed, especially cleaning, pounding, and cooking, so that we made our technologies accessories to our digestive tracts. And that's why we have very peculiar dentition and very peculiar digestive tracts. And that's the environment which we were very well adapted to for a period of about 1 million years. And then starting between 10 and 20,000 years ago for Eurasian populations, and more recently for other populations, we started to use the products of agriculture, most universally giant grass species, which are found everywhere from the New World, where it was maize, what Americans call corn, Southeast Asia, where it was rice, also a giant grass species, barley in Asia, wheat in Asia and Europe, and milk and derivatives of milk, especially cow milk, but also goat milk, in an arc from Europe through South Asia. And because of that change, we initially went through a period where our health was ruined and our stature fell and our bones became porous. And we developed a lot of chronic health problems. to which evolution by natural selection rapidly adapted us at early ages, but not later ages, because the forces of natural selection scale the speed of adaptation. So Aubrey himself was probably well adapted to a puncher's diet of beer, pork pies, Scotch eggs, and God knows what else he consumed at public school and Cambridge. But he is no longer, because he's now over 60, and the gift of adaptation to agricultural foods has left him behind. And now he's going to continue to age until the age of 90, assuming he steers away from ultra-processed foods. Inadvertently, we did the same evolutionary history with our fruit flies. We took them from being well adapted to apple orchards and the like over centuries, first in Europe, then in northeastern United States. And we gave them a completely novel diet consisting of bananas and molasses. And we fed them that diet for hundreds to more than 1,000 generations. And the evolutionary theory suggested that at early ages, they'd be well adapted to their novel agricultural-like or analog diet of banana and molasses. But at later ages, they would fare best on their long-abandoned apple diet, a diet that some of these populations haven't seen in more than 1,200 generations. And in 2020 and 2021, we showed that that was the case. That in fact, people like Aubrey and myself are too old to live on agricultural foods. We also showed that a completely novel diet to which the fruit flies were never adapted, of orange, was uniformly toxic all through the early and middle life phases. And that has given rise to my first proposed kind of intervention, which is taking advantage of this quirk of our evolutionary history, which is to go back in time over the age of 50 to what I call a whole foods emulation of a hunter-gatherer lifestyle, whole foods, 24-hour fitness gym emulation of patterns of diet and activity, in the hope of making aging stop earlier than 90, which is when Aubrey's aging will stop, and instead stopping aging somewhere in the 70s. Because a 70-year-old human who's in the condition of somebody who is Aubrey's age now, an agricultural diet, is in Aubrey's terms an eminently repairable object. Okay? So here I fully come back to Aubrey's general point of view, except that I think that the challenge of intervening right at the start, which I can't imagine ever getting regulatory approval, is replaced with a project of saving boomers like myself and soon Xers like you, Ira, and Aubrey from the continued depredations of aging can be implemented. First, stopping our aging in our 70s, and then working on any and every kind of repair and remediation project that Aubrey can imagine, or indeed that our Drosophila research might suggest, based on, a completely open-minded appraisal of what organ systems are doing.
Aubrey, the first robust mouse rejuvenation, what I'll call cassette, involves rapamycin, a senolytic, a telomerase, reverse transcriptase gene therapy, and a stem cell intervention. And then, the last time I chatted with you, I asked the question, does RMR2 add things onto that, like autophagy and extracellular matrix related stuff? Do you do a separate cassette and pass these first four onto a spin out company? And then I saw a really cool little teaser a couple days ago. You gave on Twitter that, hey, maybe once it's funded, RMR2 may have some cellular reprogramming aspect as well. I don't know if you have a scoop that you could give us while we're in this discussion, but take us, obviously, everything dependent with your work and Michael's work on lots of funding, and I hope, trillions of dollars fall from the sky for both of you. But what are you thinking about in terms of RMR2? I mean, We know
So I'll come, yeah, so I'll come to that in a moment. But first of all, there's a bunch of stuff that Michael just said, which if I don't respond to, it may leave the audience who have not been around for 20 years with some misconceptions. So one thing that Michael and I do very sharply disagree on is the idea that aging stops. Michael claimed to show, using experiments with large numbers of fruit flies about 20 odd years ago, that aging stops. But I explained in print in experimental gerontology that his conclusion was the consequence of a rather elementary mathematical error in the way that the analysis was done. And I still believe that, in fact, the data that we have, whether Michael's data or anyone else's, are completely consistent with what has historically been called the heterogeneity explanation for these things, namely that at any age you may choose, whether it's like age 105 in humans or whatever, there is a spectrum of biological age, which I would describe as amount of damage in each individual, and that translates into a spectrum of future trajectory of likelihood of death over the subsequent time. I could go into the mathematical error in question, but I won't do that because it's obviously been published already. I can always send you some stuff about that. But the key thing I want to emphasize is that doesn't really matter very much. You know, Methuselah flies are quite good enough. We don't need them to be Highlander flies. If I want to pick anything that I think is very sad about what Michael's work has involved over the past 40 years, it's the one thing that it didn't involve, which was continuation of the selection for increasingly long life. There was a hiatus, which Michael can give the actual numbers, but I think it was like a decade or so when selection was suspended. And when it was resumed with the rearrival of Kennedy Shoal in Michael's world, it resumed in abundance, and I believe that there's been an additional doubling of lifespan since then, which could have happened 10 years earlier if there had not been this suspension of the selection. So, you know, I wish that hadn't happened. And, you know, there's no sign of it slowing down, except in the sense that, of course, it inherently slows down because each generation takes longer. So this is extraordinarily exciting. I'm working with Kennedy and her colleagues at the moment, you know, identifying potentially useful experiments that one might do to pin down the genetic basis for what's really happening there. So, you know, I'm fine with aging not stopping. You know, I think another key point about this is that absolutely, we really do need to apply repair technologies to people who are 70, not people who are 90 or 100. But if we can do so, again, it doesn't matter whether their aging has stopped because we can do it every so often. And we can just, you know, if their aging hasn't stopped, which I believe, and which certainly even Michael believes is currently the case, we can't stop aging at 70 yet. Then that's fine. Rejuvenation will still work. Michael wanted to come back.
So in the late 19th century, it was becoming more and more obvious that there was something wrong with the additivity of velocities. And it started to look like there was this universal speed limit in the universe, which is the speed of light. And the physicists of the 19th century were puzzled by this. They had some fudges, which are known as Lorentz transformations, but they didn't want to re-examine their core assumptions about how space, time, gravity, mass, and energy all worked. And then this now famous dude, Einstein, then a completely unknown patent clerk in Switzerland, said, well, what if we take that seeming anomaly, admittedly under unusual conditions of velocity, and take it seriously? What does that mean about what we have to think about the relationship between space, time, mass, energy, etc.? That was the opening wedge for modern physics. That was E = mc squared, and so on. And I think the point of aging stopping is exactly the same in its import. both at the level of powerful science and at the level of technology. Because, of course, the fact that E equals mc squared is why both fusion and fission bombs are so devastatingly powerful, because they convert a very tiny amount of mass into energy. Okay? And also, that's like literally the most dramatic application of modern physics, but everybody's cell phones and computers and semiconductors, they all run on principles that derive from modern physics, as, for example, did cathode ray television sets. I think in exactly the same way, for aging, the fact that it stops in the sense that we mean it, which doesn't mean that organisms stop dying, but that in fact they stop having an exponential increase in the rate of dying, which is how aging has been demographically characterized. That is the single most devastating fact for all scientific notions which don't incorporate that idea. And it's the single most promising fact for the technologies of lifespan intervention. Aubrey wrote an article. We addressed that article and others that criticize our view that aging does in fact stop in a book called Does Aging Stop? Available from Oxford University Press online. And in fact, when we first started doing that work, we thought that this had very little practical significance. It took us, in fact, the kind of diet intervention experiments that you cued me on, Ira, to realize this was a fact of incredible significance. Because when we return fruit flies to their long abandoned apple diet, which they haven't seen in more than 1,000 generations, their pattern of aging was radically different. It was radically more benign. It made them far better candidates for interventions at any point in life, but especially mid to later in life. As a scientist, all of the fundamental requirements for the evolutionary genetics of aging are as applicable to human populations in the modern world as are true of the fruit flies in my lab. We went through a dietary and lifestyle revolution starting 20,000 years ago, spreading all around the world. Today, we still have Australasian populations in which the adoption of agricultural foods is an unequivocal disaster from the age of 8 or 9 years on. And if you return those populations to their immediately ancestral environment of an Australasian hunter-gatherer diet with some gardening, their health enormously improves. This has been abundantly demonstrated in dozens and dozens of studies. After the age of 50 or 60, we're just like them. And that's radically important because the human aging that we see on an ultra-processed food diet is a complete catastrophe. And I don't have any hope of anything in modern biology, at least for the next 30 years, remedying that. Humans on an agricultural diet are doing much better, and we have much more opportunities for keeping them alive with all the tools of modern medicine, starting in their 80s to 90s. But what's really promising is how far back, I mean, to an earlier age, we can push the cessation of aging. If we can get it to people in their 70s with the health of Aubrey de Grey or you, Ira, then in Aubrey de Grey terms, we are far more repairable. Whatever types of damage or disruption at the molecular and cell levels we have suffered to that point in time will be much more repairable and treatable, whatever those are. Whether those are, for example, the wearing out of cartilage in our joints, whether those are accumulation of amyloid deposits in our brains, whether those are any of the multiple physiological mechanisms of damage at every level, from molecule to cell to tissue to organ. All those things become much more repairable if we have in fact stopped aging at the level of health that you and Aubrey now enjoy.
Yeah,
That's the radical proposal.
Yeah, so Sherlock Holmes had something to say about this. He said, once you have eliminated the impossible, whatever remains, however improbable, must be the truth. And it's definitely very improbable that aging stops. But if one can demonstrate that the alternative that aging doesn't stop is impossible, is contrary to data, then one has to accept it. But one just has to be sure that one actually has demonstrated that the contrary hypothesis is wrong. And without trying to go into all the mathematical details with my mouth rather than with pen and paper, let me just say that I'm pretty sure that even in the book, Does Aging Stop? There was no actual analysis of my complaint about the main supposed evidence that aging does stop, namely that the analysis was done trying to fit a Gompertz relationship with heterogeneity of slope to Michael's data when it should have been a Gompertz-Macon relationship. Nevertheless, we'll come back to that in a moment. But the point is that, again, we don't actually need aging to stop at age 70 in order for repair to work. I'm not actually sure whether Michael thinks that someone who was 70 and whose aging had stopped at that point and who was then rejuvenated to, let's say, biologically 40, would continue to have their aging stopped, but it doesn't matter. Even if their aging resumes as a result of their having become biologically 40 again, we can still rejuvenate them a second time and a third time. So we're still done with rejuvenation, whether just as long as we do it comprehensively enough, whether or not aging stops. And this is why what Michael and I agree about is so much more important than what Michael and I disagree. You did ask Ira about about RMR2, and for sure we are following broadly the same design in RMR2. Of course, the design is on the website at LEV Foundation, levf.org. But we have decided that this time we will give all the mice a couple of additional things that we didn't give them the first time round. One of them is an exercise wheel, which definitely will guard against, to some extent anyway, against obesity with age. And the second one is rapamycin, which is probably the single best currently found calorie restriction mimetic. Definitely has a substantial impact on maximum and mean lifespan in pretty much everybody's hands, even when started in middle age, which is what we do, of course. So we're going to give everyone those. We might give everyone acarbose as well, because rapamycin combined with acarbose has done very well at the interventions testing program, even when started at, I think, 12 months. Or 15. But in addition, we have 4 interventions that we're going to be multiplexing in the very same way that we're doing for RMR1. And these, again, are chosen to have a lot of promise individually, but also to be very different from each other so that they address different types of intervention. You particularly mentioned partial reprogramming. And actually, that brings me back to what I was saying about calorie restriction mimetics, whether pharmacological or genetic, earlier on. It's no surprise that manipulating just a very small number of genes, sometimes as few as one, can have a profound effect on metabolism when what one's affecting is something that's programmed. We're all familiar in genetics with the concept of master control genes that originate things. So even though everyone was terribly shocked when Yamanaka was able to discover just 4 genes that we could upregulate, it shouldn't have been surprising at all. Ultimately, this is basically recapitulating something that happens in the fertilized egg, where 2 very differentiated genomes, the sperm and the egg and the oocyte, are wiped clean, so to speak, and put into a state where they can start the program. And the fact that there were only a few genes that were necessary is not surprising. It is also not all that surprising, since sperm and oocytes are so different from each other, that it works on things that it was not evolved to work on. It works on basically anything by just like ignoring what information is encoded epigenetically and just wiping it clean, almost entirely clean anyway. Mm-hmm. And that, of course, would be expected to work on any other kind of differentiated cell. So here we are with that technology. It seems to be possible to titrate it reasonably well. Especially if you leave c-Myc out, it seems that's beneficial in the adult. So we'll probably be doing it that way. But that's just one of the four interventions for next time.
And Mike, what role does epigenetics play in what you look at in 2024? Per the types of, I mean, obviously you think broadly in terms of these interventions as well, but as Aubrey and I discussed last time, and I'll bring up again now, it's clearly an area that a lot of money's getting pumped into, rightly or wrongly. Whether it's a holy grail or not remains to be seen. But does your work have, is there an epigenetic component to, when you think about what's going on in these fruit flies that are living 4 times as long, and beyond the breeding side of things, um, what does someone with your background think of epigenetics and epigenetic reprogramming as a piece of this puzzle?
So we're making a transition. This is sort of our last year of focusing on genomics, and we're really much more focused on epigenetics going forward. Literally, the transition will be taking place over the next few months. Now, epigenetics is, I have learned sociologically, now associated with DNA methylation, which is just one component of what tunes epigenetics. As I think both scientists and also clinical practitioners, we're going to switch more and more toward characterizing the transcriptome, which is the real epigenetics. DNA methylation just tunes the transcriptome. And the transcriptome is now, thanks to genomics, easily studied. It used to be very expensively studied only, but together with the declining cost of genomics, the cost of studying transcriptomes has been crashing. So our research is moving very much in the transcriptome direction. And there's something very important I would like to point out, and that is the transcriptome has become highly manipulable with the advent of lipid nanoparticles and mRNA technologies. Now, of course, that's famous in the context of the Pfizer and Moderna vaccines.
Sure.
But the people like Drew Weissman and his fellow Nobel laureate for that breakthrough, we're always thinking in terms of a whole host of diseases that could be treated with mRNAs in lipid nanoparticles. And I think that is in fact correct. I think the kinds of experiments that Aubrey de Grey is discussing with us today, which are beautiful experiments for inbred mice and dogs, are the best residues of 1990s ideas. But I think in the genomic era, we really need to think in terms of retuning based on genome-wide information, transcriptome-wide information, metabolome-wide information, because with machine learning, we can easily handle those things. They're not easily discussed on podcasts, but the truth is the undergirding of contemporary digital civilization is machine learning. And machine learning is perfectly suited to both doing the analytics on multi-omic hierarchies and even designing interventions. Basically creating things like full self-driving involves dealing with a very complex information universe and tuning your responses to it. I submit that too, in the genomic era that we've had since about 2000, is the way we will in fact best retune the physiology of humans, whether they are indeed still aging because they're under 70, or whether they've stopped at one of the 3 aging plateaus that our research reveals. I'll just add a footnote. The book Does Aging Stop extensively discusses the lifelong heterogeneity hypothesis that Aubrey and a number of others have put forward, and in our opinion, demolishes all such theories definitively. Just as the modern physicists went to a great deal of trouble to show that you got things like more bending of starlight around the sun than was predicted by the physics of the 19th century than the physics of the 20th century. The 21st century is a completely new biological era. It's not generally understood any more than the modern physics was generally understood in 1924. But of course, the events of 1945 would change that. So in a similar vein, I expect that the machine learning, or if you will, pop AI term, the AI mastery of the what you've called epigenetics, what I call the transcriptome, both in terms of analytics and synthetics, will accomplish many of the things that people are trying to accomplish now with substances like rapamycin, only far more elegantly, with far greater precision and far greater power. Because in the end, nuclear weapons put regular chemical munitions to shame. That isn't to say that there aren't still uses for chemical munitions, as the war in Ukraine illustrates. We will indeed be continuing to give people in medical clinics antibiotics and a variety of simple drugs that have good efficacy based on 20th century ideas. But the future of medicine, and especially the future of the treatment of aging, lies with the omic tools that we now have, both analytically and synthetically.
Gentlemen, you're both signatories on the Dublin Longevity Declaration. And both of you, clearly, aside from the science you do, and you do get out of the lab a lot, obviously, Aubrey, we've seen you down in Vitalia, startup city in Honduras recently, and chatting with the governor of Montana about Right to Try. Michael, you've been on the stage at RadFest a few times lately. I don't normally see you in those venues as often, but talk a little bit, both of you, if you would, about what you're doing on the public non-scientific advocacy front, aside from obviously doing podcasts. A little bit about what the scene is like in 2024 in terms of generating excitement amongst the general public, and how you feel about sort of the landscape of influencers that we have out there nowadays that are representing
Yeah,
the space.
it's been transformed. I ever since about 2007, I'm going to say, I felt that my work in terms of putting forward scientific ideas in the literature was more or less done, and that I still had a role in overseeing the implementation of a bunch of that, obviously at my various foundations. But I increasingly started to realize that I could leverage honestly, I was surprised that I was any good at it, my ability to help build the community and generally, get the word out, which essentially started to really come into its own after I was fortunate enough to speak at TED in 2006. And so I guess over that period, like those 17 years or whatever it is, I've probably spent at least twice as much of my time on the community building side as I have on the overseeing of science. And I am pretty happy with the outcome. I mean, obviously it's not been just me by any means, but an awful lot of people have come into the field as a result of those efforts, of my just, essentially saying yes to every speaking invitation and every interview invitation. And I have, if we now look at where we are, we've got exactly what I was hoping for and complaining about the absence of 15 years ago. We've got a much greater diversity of messaging out there. People who are saying more or less the right things, but they're saying them in different language. People sometimes there are I feel I'm pretty good at what I do, but I only do what I do. I say things in a particular way with a particular style, and it resonates with some audiences and not with others. And if you've got other people out there doing the same thing, but with different language, different forms of different styles, you'll reach a wider range of people. And that's what's happening now. More and more, with organizations like the Longevity Biotech Fellowship. I'll mention that one particularly because it's very influential right now. I was able to seed fund part of it a couple of years ago, and it's being run by two of the most important young leaders in the field, Mark Hammerleinen and Nathan Chang, as a way to bring people into the field who have already got past the hurdle of deciding that aging is quite a bad thing, and they'd like to, they'd like to contribute to its defeat, but who don't know how to leverage their own talents. And it's gone spectacularly well, and it has taken a lot of the weight off, that I used to be more or less single-handedly shouldering, just replying to anyone who wrote to me and saying, I'm keen on what you do, how can I help? That's the real change that's happened.
Yeah.
There's a proper community right now.
Mike?
Oh, well, everything changed with the advent of Aubrey. Uh, and he helped create not only a community of people who were concerned to do things, but also he, I think, largely on his own, generated an interest in the private sector to seriously fund anti-aging startups. When I first tried to do that um, more than 30 years ago, there was almost no one who was interested in funding an anti-aging technological effort. So the whole world owes Aubrey a debt of gratitude. I frequently compare Aubrey to Florence Nightingale, who propounded many ideas in the 19th century and was a very intelligent woman. Uh, she helped found nursing as a serious professional occupation, and she helped foster the use of quantitative methods of analyzing health risks. However, she was entirely wrong about the foundations of the epidemics of Europe. She thought they were due to bad air, miasma, and she had as a persistent gadfly Louis Pasteur, uh, who advocated instead for the microbiologist interpretation of infectious disease. And her defeat at the hands of Louis Pasteur, which only occurred long after Louis Pasteur died, circa 1920, led to modern medicine, as Peter Attia calls it, medicine 2.0, where we are very good at treating acute infection. And that's what led to the extension of average human lifespan twofold. So I am guilty of having primarily focused on the science of aging from 1975, and I will conclude my 50-year academic and scientific odyssey next year in 2025. By then, I'll be retired. I will have sent my last graduate students off into the world. I will have worked on my last scientific publications. And in the new environment that Aubrey did so much to create, I hope to finally get serious funding for ideas that are based on the conjunction of the evolutionary biology of aging, genomics, and machine learning, or AI, and that threefold concatenation or combination or synergy is what I think is going to be transformative for aging over the next 20 years. I think Ray Kurzweil is fond of predicting transformative achievements in aging in the 2030s. I hope to make him correct. I think that the continued reliance on ideas from the 20th century has been, such as Cynthia Kenyon's, which Aubrey has already criticized. I've been anchors, slowing progress. I think that in particular, given the power of omic tools to easily jump the distance between Drosophila and humans without having to worry about inbred mice as a way station along the way, will enormously accelerate progress. And many of the things that we're pioneering now with the omic and machine learning analysis of aging and Drosophila can be just moved wholesale from fruit flies to humans, because to an evolutionary geneticist, humans and Drosophila are completely parallel, unlike inbred mice, unlike nematodes, unlike inbred dogs. And we think we can make that very 21st century transition at great speed to the great, and in such a way as will greatly enhance the power and efficacy of medicine, that will greatly enhance the power and efficacy of the development of brand new kinds of therapeutics, which in some cases may involve DNA, but I think more often will involve RNA. Because the beautiful thing about DNA and RNA as focal technologies is they're information technologies, perfectly suited to the modern era of machine learning operating on digital data. Biology runs on digital data. Google runs on digital data. We basically have the spectacular opportunity to go from all of the power of the evolutionary genomics of radical life extension, which Aubrey thankfully highlighted, because it's easy for us to create far longer lived organisms with associated modifications in genome, transcriptome, and metabolome, to analyze the key points as to how that was done and start to begin the effort of developing 21st century era therapeutics based on those very powerful analytics.
One last topic I want to touch on while I have both of you. A couple months ago, the X Prize Health Fan was just announced. Did an episode with Jamie Justice on the overall structure, what's going to be happening, $100 million. Aubrey, you're on the scientific advisory board, so you can't field the team. But while we have you, I want to get just your top line on what's happening, what you're most excited about, and then I think one of the things I'm most excited about, sort of in looking at sort of the documentation, is that again, whether it's an RMR2 or whether it's a Michael Rose diet supplement combination or whatever the case may be, this prize and this unique model allows for the ultimate exploration of these synergistic combinations, combinatorial innovations, whatever you want to call them, that you guys are, working on in your own respect. Mike, I don't know if you're founding a team and you're going to be heading into X Prize, but anything that you can publicly say, please. But Aubrey, you start out as you do sit on the scientific advisory board. Say a couple of words about it, if you would.
Yeah, sure. So, yeah, I've been involved in the development of this prize since the very beginning, because actually the person with whom I founded my first foundation, Methuselah Foundation, was a friend of Peter Diamandis since before that. They actually set up their respective 501(c)(3)s in like 2001. And Peter was our first business advisor at Methuselah. So I've been talking to Peter since probably 04, 05 about the idea of maybe doing a prize in longevity. And we could just never find a formula for it that fitted Peter's intuition about having the right best of both worlds between what he likes to call achievability and audacity. And I think he was right, to be honest. It had to be a human prize. And you just couldn't, the science wasn't there to do something which fitted his achievability criterion. So everything changed around 6 or 7 years ago when, on the one hand, the science had, of course, incrementally progressed, as Peter and I had been talking on and off about this. But then on the other hand, one very important thing happened, which was that someone came along willing to bankroll the design of the prize.
Right.
X prizes are always done in a particular structure where nothing happens at all until someone puts up $500,000, which is enough to put a team together within X Prize, an internal team, to design the thing. And I never actually worked for the X Prize Foundation, but I ended up being essentially designing the prize, essentially being the overall architect from the biology side. Of course, there was all the marketing and getting the rules right and so on that had to be done. But yeah, so it was essentially put together, and as you can see, if you look at the rules, it's got my fingerprints all over it. It's basically a rejuvenation prize. It underwent a few minor changes in design over the next few years, because I basically had done all this by about 2020. Over the next few years, while Peter was out there looking for people to put up the actual prize part. But I'm very happy with how it has ended up. And there are indeed some fine details that are still being finalized. So yeah, I believe that it's absolutely the prize we need. It's a rejuvenation prize where the idea is to reduce somebody's biological age with an intervention applied over a period that has to be no more than 1 year long. And we have to rejuvenate them biologically by at least 10 years and ideally 20 in a variety of different ways, measured in terms of function at the level of musculature and also the immune system and also cognition. So it's exactly the prize that I would have wanted. And yeah, as you say, I am staying on the scientific advisory board, in contrast to some of my friends like Eric Verdon, who've stepped down from it in order to compete. I believe that I can contribute much more, given what I'm good at and what I'm not good at, when I stay on the advisory board, and I work very closely with them. So yeah, as you say, it's the biggest X Prize ever done until yesterday when they
Right.
announced $115 million dollar X Prize for desalination.
Yeah.
But that suits me fine. So yeah, I have nothing but praise for what Peter's done and what his team have done. Not least hiring Jamie Justice, whom I knew quite well already. Honestly, she's a fantastic scientist, but she is proving to be fantastic at all the things she's never been asked to do before
Yeah.
that are involved in being the executive director of one of Peter's prizes.
Mike, are you going to put together a team?
I have absolutely no funding for doing such a thing. Um, I mean, in the abstract, I would love to do that. I think, I'm retiring from the university this year. That might make it easier for me to raise the team and the funding to compete. So, I have to take this very seriously. Uh, I think it is a great idea. Um, uh, I think the 1 year time frame is a little short. I think our research suggests, because we've done a lot of switching of organisms from one regime to another, suggests that a 2 to 3 year time horizon would be more appropriate. But I mean, it's intriguing. It's beguiling. I have to retire and leave behind the ivory tower like Magister Ludi and see if this is something I can usefully contribute to.
Well, um, a lot of people said that they wanted to see a debate between you guys. We got a little bit of a debate, but at the end of the day, I mean, I think, you both, you have a lot more in common than not, and you're both making incredible, strides. You've been, over the last decades, and yeah, I called you founding fathers, which is in essence what you are. And it's a very promising period as we sit here in 2024 looking out at what's coming next. And it's just it's an honor having both of you here at the same time. Wishing you, both the best with your respective programs, Mike, on your retirement, but whatever you do next, and obviously Aubrey with RMR2 and 3, and what's coming down. Final things that either of you want to say to the audience, to each other while we have you, please. Aubrey, take the floor first, then Mike, you can wrap up after that. But again, great having both of you.
Yeah, sure. Well, first of all, thank you, Ira, for having us on the show. Every time that experts like Michael and myself are able to get out there to an audience, maybe some of the audience is unfamiliar with this work, and to, raise the quality of understanding of where we are and where we're going, every time that happens, it's another step towards hastening the defeat of aging, which is what both Michael and I are, of course, all about and have been all our lives. I just want to say, that everybody who's out there who's thinking, I like this idea, I'd like to help, you've got to look at how you can help. And that doesn't just mean the obvious things like, if you're wealthy, give money to things, if you're a biologist working in a suitable area, things like that. It also means the key thing that a lot of people overlook, namely focus on things that not very many other people are good at. This is something that people often overlook. So, in the same way that at SENS Research Foundation, I focused on the more difficult types of damage. That wasn't just because they were more difficult and therefore needed to catch up. It was also because they were more neglected, because other people were just not doing them, either because they weren't good at the right science or because their incentive structures were misaligned. It's that kind of thing. And it's not at all obvious. So I mentioned the Longevity Biotech Fellowship the other day, half an hour ago. That's a place where somebody can find out not only what they're good at, but how that can fit into the ecosystem. And of course, that's not the only example. Come to conferences. We run a conference every June, every year. This year it's in June in Ireland, in Dublin, June the 13th to 16th this year. And it's of course, a showcase of a huge amount of the absolute best science in the entire field, but also it's an enormous community thing. We very much emphasize the recreational aspect at our conferences. And so, if you're new to the field, this is the place to get properly involved, meet the right people, get to a position where you can make a contribution. So, yeah, that's what I would really say. Read up, obviously, go to our website and all the links there from. Read my book from 2007, stuff like that. But yeah, get involved. The more people we get involved, the faster this is going to happen.
Right.
Well, I'll echo Aubrey's thanks to you, Ira. It's Aubrey and I have mostly interacted live on scientific panels. I think this is our first major online interaction. And I'd like to once again express my thanks to Aubrey for completely transforming the public's view of aging, and especially for millennials and younger, converting it into a kind of crusade, and a very important crusade, much like the crusade against infectious disease in the 19th century. Aubrey referred to people doing what other people aren't doing. Well, in an era completely dominated by reductionism, certainly from the 1960s until the year 2000, I, of course, worked as a quantitative geneticist, systems analyst, and evolutionary biologist, doing what virtually no one in the field was willing to do. And in so doing, I feel that we have shown definitively, as we argue in our latest book with Kenneth Arnold and myself, that the core science on which the technology for radical postponement of aging must be based on is evolutionary biology, not reductionism. Reductionism is dying all across biology. It is being pushed into a small, tiny corner, especially involving genetic diseases and highly inbred organisms. Yes, for that minority of cases, it's as true as the moon is indeed in a geocentric orbit around the Earth, but the rest of the universe isn't. Biology is now radically transforming itself in a way that very few people understand outside of the genomics community. And this radical transformation is what will completely change our health prospects with respect to chronic aging-associated diseases in this century, and with luck, over the next 10 to 15 years. That's where I hope to make a contribution in my post-science, post-ivory tower life phase. We'll see if I make any progress at all, or if I have to wait while watching hundreds of millions of people die for reductionism.
Well, we will continue to watch both of you as you continue to champion the space. Again, for everybody going to be listening to this episode across the podcast networks or watching on the YouTube channel, again, you've been spending time with Dr. Aubrey de Grey, President, Chief Science Officer, Longevity Escape Velocity Foundation, and Dr. Michael Rose, Director of the Network for Experimental Research and Evolution, Professor, Department of Ecology and Evolutionary Biology, University of California, Irvine. Aubrey, Mike, it was an honor having both of you, and it was a great discussion.
Thanks very much.
Thank you.